So, good afternoon, everyone. Welcome to today's session. As part of this session for week 6, today, as per our usual pattern, we will be going over the topics that were discussed. First, I'll go through the assignment for the past week, where we had majorly looked towards an introduction for the cytoskeletal elements, and I will be brushing up those topics in a little more detail as well in some of the slides that I had prepared previously. Apart from that, we will also look at some sample problems as we usually do. And if there are any doubts, please feel free to interrupt me at any point of time. And I'll try my best to clarify your doubts. And if not, I'll get back to you with whatever answers I'll be able to find in the following session. For starters, we will first go over the assignment questions for week 5 that has already been uh, completed. The first question was about the plasma membrane. So micro raft domains in the plasma membrane, they are rich in which of the following elements? So there are phospholipids, polysaccharides, cholesterol, and glycolipids. In case, just so if you remember, we have had these discussion quite a few times. Also, please let me know in case my screen is not visible at any point of time. Yeah. If you remember the rafts that we had talked about, that there are certain regions across the membrane which are usually found to serve somewhat like a boat or a raft which is floating on top of a watery or on some sort of a liquid surface, and that is usually very helpful in the form that it serves as a scaffold or a substratum for certain proteins or certain elements to be localized in that position on the membrane, and they have some specific functions that they perform. Now, these lipid rafts or micro raft, as has been mentioned here, they obviously do have lipids, but what they are most enriched in, which is what gives them that specific property, are cholesterols. So cholesterols, because of their structure, because they are much bigger in structure and much because they are less fluid or they are more localized to one particular region. Therefore, the presence of cholesterol helps in maintaining that distinct region, which is quite stable across the entire plane. And therefore, they can serve as micro rafts, which have the functions that I just mentioned. Then second question was about the fungal cell wall. So we had discussed the bacterial cell wall, fungal cell wall, slight mention about plant cell walls as well, and what are the different components for each of those. For the fungal cell wall, the component that is making up the entire cell wall is chitin, and that would be the answer to it. If you remember, we had discussed about the peptidoglycans and how peptidoglycan is different in different types or different species of bacterial cells as well, and how that property or that distinction is also exploited in terms of some staining methods, which we also discussed, which was Kraft staining, for example. And we also talked about the plant cell wall, how it has a cellulose, cellulose, pectin. But for fungi, the major component that is making up their cell wall is chitin. So that answers question 2. Then in question 3, we were asked that vacuoles are dash in plant cells. So let's go through the options. We had looked at different properties of vacuoles, but among those, if we particularly talk about the options here, the first one is that said that they are membrane-bound air sacs, which is not true. Then membrane-bound protein and carbohydrate storage sites. Well, not necessarily. They do not necessarily store carbohydrates and proteins within plant cells. That's not their job. Then they are membrane-bound enzyme storage sites, also not very true. But in fact, that can somewhat be attributed to the function of lysosomes, which are majorly involved in the digestion, lysis, etc., those kind of functions, and that involves the extensive involvement. There is extensive involvement of enzymes for that matter. And in option 4, we have membrane-bound sacs that contain water, salts, and minerals, which is the correct option, as you can probably guess from here as well. This is the function of vacuoles, and we had also talked about how it helps in maintaining the turgidity, the shape of plant cells, etc. Then in question 4, we are asked that gram-negative bacteria have dash peptidoglycan layer. So we were just talking about the peptidoglycan, and if you talk about gram-negative ones, they have a relatively thin peptidoglycan layer, but they instead have an outer layer which is present the LPS or lipopolysaccharide layer, which has teichoic acids, etc. But the clear distinction between the two is determined by the thickness of peptidoglycan in gram-positive and gram-negative bacteria. So in this case, it would be thin. In question 5, microfilaments are made up of actin, myosin, alpha beta tubulin, or lipids. We had discussed this previously that the filaments, thin filaments, and intermediate filaments are also, while not completely accurate, they are also alternative terminologies which are used for these different types of cytoskeletal elements. And among them, microfilament is usually attributed to actin. We will also be going over some, not very detailed, but a little bit over these topics today as well, because there was some overlap in the topics covered in week 5 and week 6. So while you had a basic introduction to cytoskeleton, week 6 has more in-depth exploration of the cytoskeletal elements. So we'll go over that, and that would revise this thing as well. Then in question 6, we are asked that membrane lipids are amphipathic because they are dash at the polar end and dash at the non-polar end. So the amphipathic nature means that it has two different properties with respect to water, of course. So it has a hydrophobic and a hydrophilic part. The hydrophilic part is the one which is capable of or which is able to face water, to put it directly, and that is the site that is said to be the polar end as well. On the other hand, the hydrophobic part would be the side that fears water and is positioned towards the non-polar end. So option 4 would be correct for it. So hydrophilic and hydrophobic. Then in question 7, we're asked that the defect in which of the following cytoskeletal elements can lead to an inability of muscles to contract. So we talked about very briefly about it, that the actin filaments and actin as along with another type of element which is known as myosin is involved in the sliding filament theory, which is the muscle contraction phenomenon, how the thick and thin filaments, they move with respect to each other in order to allow the contraction and relaxation of any particular skeletal unit to occur. And therefore, the cytoskeletal element which is involved in the same is microfilament. So if there is any sort of defect in a person who has some sort of muscular muscular defect or muscular inabilities that the person has, it can stem from defects in their actin filaments. So in other words, those are the microfilaments, but that is not the only cause. There can be different types of defects as well, but this is the most common cause that you could attribute to it. If you're interested, try and read up on what exactly causes DMD. It is a muscular disease. It's not exactly related to what is being taught here, but you can look it up. It's a congenital disorder, and a lot of different therapies are being designed for it. Some CRISPR-based therapies, some gene therapy strategies are being looked at, and it is quite interesting. You can look it up if that's something that interests you. Coming to question 8, dynamic instability. I think we have discussed this as well. So dynamic instability is an inherent property of microtubules, actin, both A and B, or none of the above. So while you could technically answer both microtubules and actin have this property, but the inherent function of or the inherent property of dynamic instability is always attributed to microtubules because of the constant cycling that is necessary for them to possess in order to execute their default functions inside a cell. So even in actin, polymerization and depolymerization is necessary and it is required. It does occur as well. But for microtubules, it is extremely crucial for the cell to have any directionality for cell division and all of the other different properties that microtubules are credited with. Then in question 9, we were asked that which of the following cells lack a cytoskeleton, bacteria, yeast, or plants. So while bacteria might seem like the possible option. If you remember, we had discussed that there are certain homologous proteins, homologous in the sense that these are proteins that are found to be similar to the ones that you see for more advanced organisms. And that included some proteins like FTSZ, which have been found to be homologues of the cytoskeleton, and they are found in bacteria. So therefore, since none of the options are completely justified, you can select none of the above. Then coming to the last question that was part of that assignment, transmembrane protein. So since we have discussed the plasma membrane and all of the different types of proteins that can exist in it as well, and the different sort of orientations they can carry, among which some were integral membrane proteins, some were transmembrane, some were membrane anchored, lipid anchored, some were present only in one of the two surfaces. So if you remember that image, you would remember all of these different types that could be seen. And among them, the question asks for transmembrane protein. So that always means that it is, when you're saying transmembrane, it has to span across the membrane, both the inner as well as the outer leaflet. So that is option A. If you look at the other options, there would be it would either be a membrane protein, or it would be an outer membrane protein, inner membrane protein, or an anchored protein. So the correct option would be option A. And we'll get to the sample problems later. But before we get there, what I would want to now discuss is cytoskeleton. That I have some separate slides prepared. I will not be uploading these because these are a mixture of different books and taken from different sources. You can go through the video lectures if you want. But yeah, I'll just share my screen. So I hope it is visible. So basically what we'll do is we'll go over each of the elements very briefly and then move towards the more specific topics for this week. And please interrupt me at any point of time if there is any confusion. There is a lot of detail here because these were slides that I had prepared when I was studying. So these are slightly more detailed. You do not need to go into as much detail if it is not required, but just for your understanding, you can just take what is necessary for you to remember. So if you remember actin filaments, actin is made up of individual monomers, which I just get point in here. So these are individual monomers of actin, which has been indicated as G-actin or globular actin. What they do is they polymerize, they form dimers and then trimers, and they arrange in a manner which is different to that of microtubules. If you remember, I think I also have slides for those as well. But their arrangement is a little different. Their arrangement is sort of staggered. So if you have one unit like this, the monomer, then the dimer, then the trimer, they are not aligning directly on top of each other. There's a sort of twist. Much alike to that of DNA. If you have studied the structure of DNA, there is a little bit of a twist. So in case of actin as well, the trimers, they assemble in this sort of staggered manner, and what they effectively form is a filament. So while the initial actin monomer is called as G-actin or globular actin, upon getting stacked or staggered in this manner, it is called as F-actin or filamentous actin. And this happens because of addition of monomers to both ends. So what it effectively, if you remember, we had talked about polarity as well. So in case of microtubules, we had talked about the plus end and minus end. For actin, sometimes similar terminologies are also used. But in a lot of cases, what is instead used for nomenclature is a pointed end and a barbed end. So this pointed and barbed end is understood based on how these individual monomers they look like. As you can see, one end is a little more pointed and the other is sort of, it has a little cleft. So that clefted side is the barbed end and the other pointy side is the pointed end. So that is how an individual actin filament would look like. So next, what is being mentioned is that how these individual actin monomers, how each monomer is getting added to form that elongated stretch is very heavily dependent upon how enriched or how concentrated or how abundant the monomers are within the cell at one particular point of time. So there is a critical concentration. Again, this is quite detailed, but there is a a minimal concentration that is required, at which the rate of polymerization into the filament, the rate at which they are getting added, is equal to the rate at which they are getting dissociated. So if you have like imagine imagine little pasta, right? If you know, if you have seen penne pasta or any sort of pasta which is commonly available, and you keep on adding them one after the other, and you keep one person keeps on adding them, another person keeps on removing them from other end. So the rate of this addition and dissociation, when it is equal, then these are said to be in equilibrium. The availability of monomers inside the cell and the filaments which are present inside the cell are said to be an equilibrium, or it is, you can consider it to be the homeostasis, the condition which is known as homeostasis. The two ends, the pointed and the barbed ends, they grow at different rates, and the barbed end, which we talked about, which has that little cleft, has a much faster rate of addition, about 5 to 10 times, as you can see here, around 5 to 10 times faster addition occurs at the barbed end compared to the pointed end, which is considered to be much slower growing. And how this process actually occurs is something that is quite uh, different, you can say, compared to like this point of initiation of actin filaments versus that of the microtubules. Is it has a major point of distinction in the initiation part itself. So for actin filaments, the initial step and also the rate limiting step is how this nucleation is occurring. So this step, which is known as nucleation or the initiation, it is there's a particular terminology which is said to be ab initio, which means it can start from scratch, from nothing. So monomers are able to interact and they are able to form this entire filament. For microtubules, the situation is quite different, and we'll get to that. So there are certain proteins, not getting into detail about it, but for example, there is furin and there is formins and there are ART23 complexes. It was briefly mentioned there. For now, you can just look at it here that this is the formin dimer, sort of similar to the structure of ribosome, if you remember, that acts as a scaffold or a stage on which the individual actin monomers are attaching. And these monomers, they keep on forming trimers and they keep on assembling, and that entire stretch of filaments can then be formed. Just for your understanding here, 6 such monomers have been shown. So you can see that in the order of addition, 1, 2, 3, 4, it is getting added and it is eventually forming forming the F actin or the filamentous actin that you are more commonly familiar with. And another protein, which is ARP23 complex, it determines where the filaments can be formed within the cell by these initial filaments. Now, this is the process via which you can get an elongated stretch or just long, just the filament-like structure of actin. However, because like we have previously discussed, and as you can see in the slide has been mentioned, just give me a minute. Sorry for the disturbance. So yeah, I was saying that when you consider the function of actin, since it is very closely associated with the plasma membrane, it is localized just beneath the plasma membrane. It has a huge role in the movement of a cell, or in directing some region of the cell forward or backward, and throwing out the cell processes that we talked about previously. So at this leading edge, actin filaments need to undergo this process, and particularly this process of addition and removal, it is often also called as treadmilling. The concept of dynamic instability and treadmilling, why treadmilling is, if you just think of the if you think of anybody who is walking on that treadmill and you think of it much like a chain, sort of the chain of any bicycle that you have, or like a conveyor belt sort of a structure. So you keep on walking and you see that from one end, it is getting much analogous to what I just explained about the pasta analogy, right? one end is getting detached or dissociated, at the other end something is getting added. So there is sort of a cycling motion or a treadmilling motion that occurs. So at the leading edge, or on the ends where a cell is moving in a particular direction, it has to have a lot of active treadmilling for actin filaments that must occur, but that would not be enough. If it has to move further, it needs to branch out. This branching is quite heavily taken care of by the ARP23 complex, which we just mentioned, the actin-related protein. Yeah, here, actin-related protein, or the ARP23 complex. So what it does is, you can see that this green colored protein, it is associating at some particular region along the length of the actin filament, and it is redirecting, not necessarily redirecting, but yeah, the correct term for it is branching out. So the normal forward growth of the filament is also taking place, which is why you're getting an extension in that direction as well. But there is also an extension in a different direction, which is sort of branching out the entire cytoskeleton for the cell process to be formed. So this is how the process occurs. And then you can see that regulation of this actin filament is not just taken care of by these two proteins, but there are other proteins as well. Here also there's a lot of detail. Don't get intimidated by so much detail. I'll just mention the name of the proteins without going into much extensive details about it. But like we just saw, the initiation of the filament and the polymerization step, that is taken care of by certain proteins like formin and ARP23. Then, after the filament has been formed, it is stabilized by some other proteins, nebulin, tropomyosin, etc. Then comes cross-linking. In actin, there are different sort of structures also that can be formed. Like there are some actin bundles which can be formed. Actin mesh sort of structures can be formed depending on what proteins are formed. So you are getting, think of these as individual filaments. When these filaments are stacking in a particular manner, such that a cylindrical sort of structure can be formed, then that is an actin bundle. It has a different role. Versus if you have crisscross actin filaments like this, which are just forming a mesh-like structure, that has a different job. So different types of proteins are responsible for different types of cross-linking, which is taken care of by proteins like alpha-actinin, filament, fibrin, vilin, etc. Then there is a capping of actin which occurs, which is shown here. So capping proteins can often be CAPZ or tropomodulin. No need to remember, just to introduce you to these topics. Then there is a filament severing or depolymerization. So severing basically means you are cutting or severing or just chopping the filament into two or into multiple parts. So that is getting severed by proteins such as cofilin or ADF, gelsolin, thymosin, etc. Then comes binding of monomers, which profilin takes care of, etc. And then there is linkage of the actin filament to other proteins, which is taken care of by proteins like dystrophin. This you will find to be quite relevant to DMD, which I just mentioned. There is there's something very interesting about dystrophin as well. You can look it up. It has some specific uniqueness about its expression in the human body. You can look it up. It is interesting, and it is one of the majorly studied disease in the world today, apart from neurodegenerative diseases and cancer. So you can look it up. It is interesting. So these are all of these different types of proteins which associate with the actin filament for different types of function. Like you can see here, there is polymerization, depolymerization. Different proteins are involved in that. Some are involved in cross-linking, some are involved in severing, some are involved in capping for different types of functions. Now, coming to the protrusions that we talked about, that for a cell to move, actin has a major role to play in that. And this is taken care of by some sort of specific structures, and I'll get to them very briefly without getting into detail. You were already introduced to these concepts in the lecture, so I'm not unnecessarily detailing those. So among the actin-based protrusions that are usually associated with that are usually associated with these functions, the best studied are microvilli. So what are microvilli? They are finger-like extensions of the plasma membrane, and they are abundant on cell surfaces involved in absorption, which, of course, the most common example that most of us are familiar with is within the intestinal lining or along the intestinal lining, where you would find them to be very extensively present. And these brush border epithelial cells are involved in many folds, like 100-fold increase in the surface area of absorption during the process of digestion is mediated because of the presence of these microvilli. And what they form is known as the brush border, and it has 1,000 microvilli per cell, and the exposed surface area, I said 100, sorry, it's 20 folds increase. The exposed area increases by 10 to 20 folds. And if you remember, we had seen a bacterial cell sort of cross section in the last week, and there also be some villi which are present on the surface. So while they do not have the same purpose, but villi are also present on the surface of the cell, they are also protrusions on the surface. And do not confuse between the two, because bacterial cytoskeleton is something that is still being explored. It is not made of actin. I'm not trying to draw the analogy there. What I'm just saying is that it also has a similar function in terms of increasing the absorption, just for that reference. In contrast to microvilli, there are certain, so while microvilli, as you understand, they are, you don't lose, or you do not have a sort of turnover of microvilli inside the human body, right? They are constantly present and present within an individual throughout their life. There can be changes based on an individual's age, that, with less food consumption or more food consumption, the need for digestion to be accordingly adjusted. Or if a person suffers from some other disease, then there would be a difference in the expression or the presence of microvilli. But in general, their presence is a lot more necessary, a lot more constant in comparison to other structures, which is what we'll be coming to next. So pseudopodia, lamellopodia, these are the two other types of structures which are considered to be much more transient, and their formation is more related to the environmental stimulus. So pseudopodia, they are extensions of, they are also extended or projected structures of moderate width, and they are made up of actin filaments that have been cross-linked into a 3D network. They carry out phagocytosis, and they allow the movement of amoeba across the surface. So if you have, I think we all have done this in very young age, sometime in high school, that whenever you drew an amoeba, you basically drew a very haphazard structure with a little nucleus in the center. And those individual extended regions are usually what we understood as pseudopodia. So these pseudopodia, they are said to be made up of actin filaments, which are cross-linked into a three-dimensional structure. Please let me know in case my slides are not visible, because I'm trying to share my screen only. I'll just share it once again. So we were talking about pseudopodia, and then come lamellopodia, which are also projections, but they have a difference in the morphology compared to pseudopodia. So they are more broad, more sheet-like extensions at the leading edge of some specific types of cells, which are fibroblasts. In case you're not familiar with them, you can skip that. Just know this for your own understanding, that fibroblasts usually have these structures, and they are also made of such actin filament networks. Many cells also have philopodia, which are microspikes, and these are much thinner projections, which are of the plasma membrane, which are supported by actin bundles. And the extension, retraction of these structures based on how the actin filament is getting assembled or disassembled at that particular region, and that is what kind of what we understood by the process of treadmilling as well. So we looked at majorly four structures. There is, just to revise it again, we saw microvilli, pseudopodia, lamellopodia, and philopodia. So these are the four major actin-based protrusions that are present on the cell surface. Now coming to the next type before we get to microtubules, because we had not really gotten into any detail about intermediate filaments. So IFs, or intermediate filaments, they are, as the name indicates, these are, their dimensions are somewhere intermediate between those of the other two common cytoskeletal elements. So while it is somewhere 10 nanometers in diameter, it is extremely important and has the major role of providing mechanical support to the cells or to the tissue at large. So they have an approximate diameter of about 10 nanometers, which is intermediate between that of the two other principal cytoskeletal elements, which are actin filaments, somewhere around 7 nanometers, and microtubules, which have somewhere around 25 nanometers. So if you remember, there was a comparative table in the previous week slides as well. So you have thicker, intermediate, and thinner filaments, and their nomenclature has also been done in a similar manner. Then we're talking about the third point. So in contrast to actin filaments and microtubules, intermediate filaments do not play any major role in any sort of cell movement. Instead, their role is more mechanical in nature, more structural in nature, that it is serving as a support system or infrastructural or architectural element for the cells and for the tissue at large. And they're not just made up of one type of protein. They have a huge range of variety of proteins that are expressed in different types of cells. You'll find a wide range. Some are known to be acidic, basic, and you can look at the different types. There is keratin and there is pimentin, there are lamins, etc. So there are different groups, intermediate filament groups, and majorly they have been classified into six groups based on their similarities and differences. I'm not going into detail of it, but because we were talking about how each of these cytoskeletal elements have been they get assembled, and we saw that how actin gets assembled and how there are so many proteins that are involved in just the assembly of actin filaments, that if you start thinking from the initial point, then you have to understand that there are genes for actin, that its gene expression is, first of all, that gene has to be copied correctly during the process of replication. It has to be transcribed, translated, and then you get that individual actin monomer. That actin was getting formed into trimer, that was forming a filament, that filament was getting branched, and then it was forming sheets and meshes, and then it was associating with the plasma membrane and then helping a cell to move. So there are so many processes that are taking place in absolute coordination and with so much accuracy for just one movement of the cell to take place. It is incredible if you think about it. So that was just one sort of assembly that we understood. But when we talk about intermediate filaments, then in their case, you have to see that there's some difference in it. First, if you remember, we had mentioned this in the previous class as well, that they lack polarity. Now, why do they lack polarity? I had shown you just like with my hands, but I have a better schematic for you to understand it today. So first step is that the individual, here what you see is a polypeptide and it has an individual filament, one filament. Now, what it first does is that it forms a dimer, and it forms a dimer in a coil manner. So the filament initially has an N-terminal and a C-terminal, N-terminal head, C-terminal tail, and the center has a rod-like domain, which is the polypeptide or the protein. This forms a coil structure such that the N and C-terminal region, or the directionality or the polarity, is still maintained. Then these individual dimers are forming a tetramer. These tetramers are forming a protofilament. And these protofilaments are all assembling around one another to finally form the filament, which is much structurally quite similar to that of microtubules, functionally obviously not the same. So this was what you understood from the schematic. I'll just go over the writing as well for your understanding. So the first step is there has to be formation of dimers, where the central rod domains that we saw, they would wind around each other in a coil structure. Then they form a staggered sort of arrangement, staggered as well as anti-parallel, and this step during the formation of the tetramer, where they are becoming anti-parallel to each other, this is where they are losing their polarity. That since you see that the N and C terminal are on the same side for each of these two, it is negating the polarity that they initially were carrying. If this was constantly maintained, then even intermediate filaments would have some sort of polarity. Then the final intermediate filament, it contains approximately 8 protofilaments, which have wound around each other in a rope-like structure, and they are equivalent, so they do not have any polarity. And in contrast to actin filaments and microtubules, these are said to be apolar. They do not have distinct plus and minus ends. Not just that, they also do not have that treadmilling or dynamic instability that we saw in those cases, which is why you cannot really determine the cause and effect in that case. But because they have a role to play in the cell movement, And because that is a necessity for those two, the other two cytoskeletal elements, there has to be treadmilling and there has to be dynamic stability, instability in their case. Versus because the job of the intermediate filament is mainly structural support, it does not need to have those properties. And so it lacks such treadmilling and it forms a strong, stable structure inside the cell. Now we'll come to microtubules, and from microtubules, we'll diverge towards the motors, the molecular motors, and then we'll just briefly go over the lecture notes as well, because it has quite overlapping details in both cases, but I just felt there were better schematics here for you to understand, so I included them. Okay, so first of all, microtubule, the structure. What are they? They are rigid, hollow, rod-like structures, approximately 25 nanometers in diameters. They undergo constant assembly and disassembly, and they function, one of their major functions is in order to determine the cell shape. So there are a variety of cellular movements that include some sort of cell locomotion, the transport of organelles inside the cell. If you remember the video lectures that were shown, that the molecular motors, they carry whatever cargo is inside the cell from one location to another. Example being neurotransmitters for neuronal cells, where from the postsynaptic neuron, from the presynaptic neuron to the postsynaptic neuron, whatever neurotransmitter has been released, that has to be taken from cell 1 into cell 2, and that has to be taken along a railway track, which is majorly made up of microtubules. So these are their two major functions. One is cell movement. The second is intracellular organelle transport. And the third, which we have not gotten into much detail, but is a very interesting topic. And if there is enough time, I don't think there will be, but if there is enough time, I will go through that because it's extremely relevant, extremely important as well. So these are the major functions. Now, what are microtubules made up of? They are made up of a single type of globular protein that is known as tubulin. These tubulins, they are consisting of individual dimers that have two very closely related polypeptides, 55 kD. If you remember, we had talked about housekeeping genes as well. So I had mentioned it in the previous class, which are used as controls in techniques like Western blotting. So these actin is somewhere around 45 kilodaltons. It is usually, you get a band for Western blotting for actin at around 45 kilodaltons, and for tubulin, beta alpha beta tubulin is somewhere around 55 kilodaltons as well. So if you are checking for a high molecular weight protein, you would, it is easier to visualize compared to overlapping proteins. Those are technical details. We don't need to get into it. Coming back, alpha and beta tubulin, they are related, or they are encoded by quite related genes, small families of related genes. But in addition, there is another third type of tubulin as well, which is known as gamma tubulin. And its job is very, very relevant to microtubules, but it is not a component of the microtubule by itself, but has a role to play. Where it has a role to play, something will come to. It is concentrated in the centrosome. And in the centrosome, why is it so relevant? The centrosome is the site where microtubule assembly is initiated. Another point, so this is where you need to, whenever you study cytoskeletal elements, and because these are so similar and yet so different, they are not redundant in nature. They are quite similar, but they have very defined separate functions. So while we saw that actin synthesis, or not synthesis, actin nucleation can occur ab initio, can occur by itself. Of course, you need some assisting proteins, but it can start from nothing. It can just start from assembling some actin monomers. In contrast, for microtubules, you need to have an entire setup in place. And that setup, it starts at the centrosome. That is where it has to take place. So in the centrosome, the gamma tubulin, the third type of tubulin, is very, very concentrated, and it is credited with the job of microtubule assembly as well. And here you see that the evolutionary ancestor of all plant as well as animal tubulins, they appear to be a protein which is very similar to the prokaryotic protein, and that is known as FGSC. So this is a schematic representation, as you can see, of how a microtubule is, what it looks like. So if you just see its like top view, you would see that it has an inner diameter of around 14 nanometer and an outer diameter of somewhere around 25 nanometer. And you can very easily calculate the radius of each tubulin unit. But you have to keep in mind that inside the cell, they are not individually made up of spheres. These are schematics, and therefore they are not perfect. Tubulin is not a perfect round sphere. It is a protein, so it has a specific orientation which it holds. So just out of curiosity, you can try it out. But this is what the top view looks like. And from the side, you can see that you have these individual alpha beta tubulin dimers, which are associated not in a parallel manner. These are also staggered, much like actin, and they have this sort of twisted orientation that you can see. So tubulin dimers, they polymerize to form microtubules, and these have 13 linear protofilaments that are assembled inside a hollow core. So that is what you see here, that the core is hollow and it's approximately 14 nanometers in diameter. Um, the protofilaments, which are composed of head to tail arrays of tubulin dimers, they are arranged in parallel. Consequently, the microtubules, much like actin filaments, are polar structures that have two distinct ends. There's a plus end and the minus end. Now, the very direct interpretation of the plus and minus end is that the plus end is where addition is occurring, minus end is where subtraction is occurring. So the plus end is said to be the fast growing end, and the minus end is said to be the slow growing end, similar to the barbed and pointed ends that we talked about in case of actin filament. So this polarity is something that is very important in determining the direction of movement along microtubules. That if there is a certain protein and it has to move towards one of the two directions, it has to either move towards the plus or minus end. That is a major factor that has to be considered. Okay, now, excuse me. Now, tubulin dimers, they can depolymerize as well as polymerize, and microtubules are capable of undergoing very rapid disassembly-assembly cycles. So, what you also need to keep in mind is that the energy source that is exploited in the assembly and disassembly of each of these cytoskeletal filaments is different. In case of actin, we saw that there is ATP, but in case of microtubules, it is GTP. So alpha beta tubulin, they bind GTP that has similar or analogous function to that of ATP, and it regulates polymerization. The GTP bound alpha beta tubulin undergoes hydrolysis, forms GTP during polymerization steps. And based on this, there is a depolymerization which occurs, and that favors the dynamic behavior. No need to get into it. Just understand that there is a dynamic instability which is maintained because of the cycling that occurs between the tubulin subunits and GTP or GTP bound bonds. Um, I think I can skip it. I'll instead show you the schematic representation of what treadmilling and this entire dynamic instability process it looks like. So here is the plus end, and you are seeing that the GTP bound monomer, I'm not sure if it is clear here, but yeah, these are GTP bound monomers which are getting added to the plus end of the microtubule, which is the fast growing end. And you can see that there is dissociation which is majorly occurring because it is a slow growing end. More dissociation is occurring at the minus end. So the minus end, it is, I'm reading out the legend or the caption. The minus end, it grows less rapidly than the plus end of microtubules. And this difference that you see in the growth rate, it is quite heavily reflected in the by the difference in the critical concentration for tubulin dimer addition to the two ends of the microtubule. So tubulin dimers that have GTP bound tubulin, they associate with a plus end in a flat sheet-like manner, and then it zips up into, if you can see here, these are assembling in a sheet-like manner, then they're zipping up and they're forming the hollow microtubule that you're familiar with. And after this GTP bound beta tubulin, it has polymerized, it undergoes hydrolysis. And here you can see there's a change in color from that orangish to this reddish color, which is where GTP hydrolysis has occurred. GDP has been broken down and GDP is now present. So this GDP bound tubulin is relatively less stable in the microtubule, and therefore the minus N tubulin units, they begin to sort of dissociate or they begin to peel off. And treadmilling takes place at the tubulin dimer concentrations, which are intermediate between those for the minus and plus end, and there is a net dissociation from minus end balanced by the addition of dimers to the plus end. So this is what how the growth and or you can think of it as the addition and subtraction of tubulin dimers to the existing cytoskeletal microtubule units can shorten or lengthen one particular region inside the cell. So this is how that process occurs. And this dynamic instability, this terminology, if again I would heavily urge you to read up on this topic because it is a very not very, but yeah, certainly a complicated topic to understand, but it is very crucial towards clearing of your concepts for how inside the cell things are not just static, things are not just moving about. Everything is dynamic, and so many things are coordinating in order to make anything feasible. Like for example, this dynamic stability that we see, it has such an immense rate, such a quick rate of few minutes. Like if you see here, just half-life of a few minutes are present, and this rapid turnover that within a few minutes, one particular region of the cell can look completely different from a few minutes later. So this is very critical for the remodeling of the cytoskeleton that occurs during mitosis. And this particularly is exploited. I think I had mentioned this in the previous class as well. There are certain drugs which target this microtubule assembly, and they are not just used for cell biology understanding for experimental techniques, but also as cancer treatments. So you have colchicine and colsemate. They are examples of very commonly used experimental drugs. They bind to tubulin and they inhibit microtubule polymerization. That plus N addition would be stopped. So that in turn ends up blocking mitosis, which is something that is very, very useful when you're treating a cancer patient. But yes, these are not the first lines of treatment. They are not something you immediately give to the patient because there is very less specificity. So what it means is that when a particular patient is consuming colchicine as a drug, as a medicine, then that individual within that individual's body, these drugs will go and work for each and every cell or can work for each and every cell within the body, right? So they would not necessarily or specifically target cancer cells, which would lead to a lot of off-target effects or unwanted effects in those cases, which is of course not something you desire. In fact, that is something you do see in chemotherapy as well, which is why patients that undergo chemo, they usually suffer from hair loss, from their head, from their eyebrows, and because that effect is non-specific, it compromises their overall health, their immunity. So these off-target effects are something that you eventually move towards when the treatment is at a stage that what is already known to mankind or what is already known to medicine is no longer working for such a patient. And all you want to do is allow them to survive or try your level best to help those patients survive for as long as possible. That is known as palliative therapy. So Taxols, which is another class of drug which I'll be coming to, are given towards these extremely end stage disease where you know that none of the other preliminary treatments would be useful or they have already been tried out and they have not been useful. That is when you resort to such sort of drugs. So you have colchicine, colcimate, you have vincristine and vinblastine. These are relatively more specific because they inhibit the rapidly dividing cells, but they also have their cons associated with them. Um, if it is interesting, you can read up on those. One of the major struggles in cancer biology is figuring out the specificity. It is trying to personalize medicine, but also trying to cut down on the cost so that you don't have to synthesize a new medicine for each diseased person. But yes, that is also being generalized. If you've read upon CAR T cell therapy, then that is a concept which is being explored in that field. So there will always be new questions and there'll always be better solutions that come up. But yeah, the basic concept that is exploited in these cases is just stopping the assembly and disassembly of microtubules in their case. Taxol, like I said, Taxols, instead of inhibiting assembly, what they do is they stabilize the microtubules, and this stabilization effectively ends up blocking cell division. So these are examples of some of the drugs that are found to be quite relevant to cancer chemotherapeutics, but they are also not the most effective, but they are what has already been studied. So I now coming to the assembly of the microtubules. So like we talked about the fact that ab initio synthesis of microtubules does not occur. Instead, the centrosome is the uh, the anchoring sort of region inside the cell, which helps in this intracellular microtubule organization. So in animal cells, most microtubules, they extend from the centrosome, from the microtubule organizing center or MTOC, and they are projecting outwards or extending outwards. And here you can see that the centrosome is very closely spatially very closely associated to the nucleus, that it is located right next to the right adjacent to the nucleus, near the center of the interphase or non-dividing cells. That's what you can see here, that this is the you can see that this is the nucleus, and very, very close to it in very close proximity is the centrosome. And from there, you can see the outward projections. Keep in mind that here you can see there's a difference in the orientation that is visible. So it is during the process of mitosis, you can see that microtubules, which are extending outwards from duplicated centrosome. This is a mitotic cell where a duplication has already occurred. And in such a cell, you can see that there are two centrosomes instead of one, because that will also get divided between the two daughter cells. And obviously, you do not see a nucleus, because during the process of mitosis, what you want is that the genetic material should be equally distributed among the two daughter cells. And there you can see that the genetic material, which was previously inside the nucleus, has undergone disassembly and has sort of organized itself, has condensed itself in the form of chromosomes. And those chromosomes are aligned or are arranged along the metaphase plate or along the same plane, from which separation in two different directions would take place. So during the process of mitosis, these microtubules, they extend outwards from the centrosomes to form the mitotic spindle, and this helps in separation and the distribution of chromosomes to the daughter cells. Now the centrosome functions as a microtubule organizing center, in which the minus ends of the microtubules were initially anchored. And it serves as the initiation site for assembly of microtubules. They grow outwards from the centrosome towards the cell periphery. And this initiation of microtubule microtubule growth, microtubule assembly at the centrosome helps in establishing the polarity that we talked about, that towards the microtubule, the since microtubules grow by the addition of tubulins to their placents, which are extending outwards, the placents project outwards from the centrosome towards the cell periphery. The centrosome initiates the microtubule growth, and that is where the minus end is initially anchored. From there, assembly has taken place, and more assemblies occurring towards the placents as it is extending in that particular direction. So particularly, this can be very clearly seen or visualized in cells that have been treated with colcimid or colchicine that we previously saw, that assembles the microtubule. As long as the drug is present, you will see some sort of disparity which is taken care, which is where this assembly is occurring. The entire assembled microtubule is getting dissociated. And when you're removing that drug, the cells would be able to recover and new microtubules would grow from the centrosome again. Within the centrosome, the key protein, as we saw, is gamma tubulin, and this gamma tubulin is the major species that nucleates the assembly of microtubules. Now, within the centrosome, what you saw here was a green sort of cloudy, hazy sort of a structure, which has something to tiny things present at the center. What are those things? What is inside the centrosome? So inside the centrosome, they have centrioles, and centrioles that is surrounded by that hazy sort of mass that is known as the pericentriolar material. So that's what you see here. There are two centrioles which are oriented in perpendicular orientation with respect to each other. And the surrounding hazy cloud-like material that you saw, which is amorphous in nature, is the pericentriolar material. Now, the individual centrioles, representation of which has been shown here, they are also cylindrical structure. They also are made up of microtubules, but these are not necessary for the microtubule organizing functions of the centrosome. What is important is what is present inside, that is gamma tubulin. So towards the outside, what you're seeing are these sort of panels of three microtubule units, the alpha, beta, and delta tubulin. And on the inside, if you think of it like Again, the analogy of pasta. So if you're making some sort of say Alfredo sauce pasta, then what is on the outside, the shell itself is made up of microtubules, but whatever gravy or whatever Alfredo sauce you're getting on the inside, that would be the gamma tubulin. That is from where the nucleation of new microtubules would take place. So it is this along with the pericentriolar material which is initiating the microtubule assembly. However, if you remove centrioles from animal cells, I'm not just saying that gamma tubulin is the only region. This, along with the pericentriolar material, together help in microtubule assembly. And if you remove the centrioles from animal cells, then it would disperse the centrosome contents, and microtubule turnover would be significantly affected. That you would get much less microtubules, basically. So, till now, we have looked at each of these filaments, their assembly, what is the difference between them, their orientations, their contributing factors, organizing centers, etc., etc. Lots of details about those things. What we will look at now is the involvement of microtubule motors and the movement and why it is necessary. Okay, so microtubules are responsible, as we've already discussed, for a number of different types of cell movements, and that includes the transport within the cell, which is intracellular transport, for the localization of membrane vesicles and organelles, for the separation of chromosomes at the time of mitosis, and for cilia and flagella beating. So what exactly is conducting, or not just conducting, but also regulating the function of or the activity of movement along microtubules are the motor proteins. What they do is they utilize the energy which you are getting from ATP hydrolysis, and they're producing force and movement. There are two families of these motor proteins, which you're already very familiar with. One is kinesins, the others are dyneins. Kinesins and dyneins have opposite sort of movement. The directionality of movement is a topic that was quite extensively discussed by Professor Subramaniam in the lectures as well. It is it is something that you can go and read up on. I'm not familiar with the most recent research about it, but it is something that is being explored. That how um, relevant, or how, what should I say, how fine-tuned is the directionality of these molecular motors important within the cell? What are the pros, what are the cons of the movement, if it is unidirectional versus bidirectional? How is it made? So what is the determining factor? Why can't it switch that at some point of life based on some sort of stimulus? So there are lots of open-ended questions, some of which might already have been published. You can look it up if you want. But for now, the general understanding about these motors, molecular motors, are that most kinesins, they are, since they are not just like, this is another reason why generalization of the function of these molecular motors is quite difficult. That is because kinesin is an entire family of proteins. Dyneins are a separate, entirely different family of proteins. So among them, most kinesins, they move towards the plus end, the ends that are projecting outwards or that are extended outside of the centrosome, versus dyneins, which are moving towards the centrosome. So they are moving towards the minus end. The kinesins are plus end anchored molecular motors, whereas the dyneins are minus end anchored molecular motors. So here you see a schematic for that as well. This is what a kinesin looks like. This is what dynein. These are again representative images. They're made up of individual chains that have all like coiled around each other and made up the 3D structure. So you have kinesin 1. Kinesin 1 has two heavy chains, two light chains, and you can see here that the heavy chains, they have long alpha helical regions. They are wound around each other in a sort of coil structures, and you have a very globular heavy N-terminal head domain of the heavy chains, which are the motor domains of that molecule. They bind to microtubules and to ATP. And these head domains, what you can see here, obviously. If you remember the initial video, I think I had separately also mentioned it, that how do these molecular motors move along the cell. So you can think of them as feet, and these motors, as they're moving along the microtubule, as they're moving along the microtubule, they carry out ATP-GTP hydrolysis, and that provides the energy that is needed for this movement to be executed. On the other hand, the tail portion of the kinesin molecule that consists of the light chains, which are associated with the C-terminal domains of the heavy chain, and this is responsible for binding to other components. So just to clarify it, what you see here, what is attached to the microtubule is the head domain. What is far away from it is the tail domain, and that is where it carries the cargo. Think of it as a person who is carrying something on their head. The cargo is carried on the head of these molecular motors while they are walking on the microtubule. So I think if I just flip the diagram, it might have been easier for you to just visualize it, but this is how it exactly occurs inside the cell as well. And the arrows that you see here are indicating the directions in which they normally move. Again, there is a lot of subclassification under it, but kinesins are shown to move towards the plus end, and the dyneins are shown to move towards the minus end. And talking about the cargo that they carry, that can include other cellular components like membrane vesicles, like organelles, and these all are carried out by the action of the kinesin motors, because as you understand, if something is getting directed towards the centrosome, it is not necessarily useful, right? If it has to be localized at some specific position within the cell, then it has to move along the microtubule from the minus end towards the plus end. It has to go and localize somewhere inside the cell, and that has to be taken care of by, or that is usually taken care of by the kinesin motors. Uh, in addition, cytoplasmic dynein, which you can see here, is an extremely large molecule which has 2 or 3 heavy chains, number of light chains. And the heavy chains, they form the ATP binding domains, which are helpful for the movement along microtubules, the head domains that we talked about. And the basal portion of the molecule, which has light as well as intermediate chains, binds to other subcellular structures like organelles and vesicles, which we talked about. And it also, not important for you guys, but yeah, it also can also interact with some proteins like dynactin to move over very long distances along the microtubule. So depending on what is the job, these orientations would be different, and they would have different functions. They would have different, for lack of a better word, collaborators or coordinating proteins, or something similar to that of cofactors, not necessarily cofactors, they're also proteins, but yeah, some other molecules which help or which supplement the job that was already being done. Here, this has a lot of detail. You don't really need it. Just we'll go through it really fast, but this is what the cross-section of a cilia it can look like. It is the axoneme, which has microtubules and their associated proteins. So this is a cross-section of an axoneme. What you see here are that there are 9 doublets which have one complete and one incomplete microtubule. If you see here, this is the incomplete and complete microtubule. Too much detail, but just to show you that this is how arrangement occurs inside the cilia and flagella. So there are 10 or the microtubule that has only 10 or 11 protofilaments, and the outer doublets, they are joined to each other but by certain proteins. The green, I think the green ones, if I'm not wrong, yeah, the green proteins which are known as nexins, and the arrangement that you see here, that is usually known as the 9+2 pattern, which has a central pair of microtubules. This pair of microtubules, that is the 2, and surrounding there are 9 outer microtubule doublets, and these doublets are incomplete. These doublets are distinct. The 2 fused microtubules of each outer doublet are distinct. One is called the A tubule, which is complete and has 13 protofilaments. The other is incomplete, has 10 or 11 protofilaments, and they are connected to the central ones by radial spokes and to each other by nexin. And dynein, 2 arms of dynein are attached to each A tubule. And here, these blue proteins that you see, they are dynein arms. Just for your understanding. So you have dynein here shown in blue, which are getting attached to the A tubule, and these axonemal dyneins, they help in movement of cilia and flagella, because you know that particularly for flagella, it has a huge role in cell locomotion. So it has this sort of beating structure that you usually also see for fish, right? That's how they swim. So this beating structure has, or this beating movement is mediated by the motor activity of these axonemal dyneins, which is why I at all went into this cross section of what an axoneme looks like. So yeah, this is what you see here. This is what Again, too much information, but just if you're interested, what you're seeing here is that within the cilia and flagella, the minus ends are anchored in sort of a basal body, and it is similar to a centriole. It has 9 triplets of microtubules. What happens is that the outer microtubule doublets, they slide along one another, and that is mediated by dyneins. You can see such tiny multiple dyneins which are present between these two doublets, and as the dynein head group, it moves towards the minus end, which is the direction in which it is moving. Then what it does is there is some relative movement with respect to each other. It causes the A tubule of one doublet to slide towards the basal end of the adjacent B tubule. So there is a movement which is occurring with respect to each other. And since they are all connected by Nexin links, the sliding of one doublet causes another one to bend, and therefore it results in an effective movement. If it is too confusing for anybody, just keep in mind that the dyneins that are present between each of these two doublets, they move along the direction, along the minus end or towards the minus end. And there is a relative movement or relative sliding which is occurring between the two doublets that is resulting in that beating sort of a motion that you see in cilia and in flagella. So that kind of covers the slides that I had prepared. We will now come back to some of the questions, I think. Um, Maybe we can just, yeah, I think I'll go through the sample problems first, and then we can just go over the lecture notes that are already available to you. So, if you remember, we have just, based on what we have discussed just now, the first question is that microtubule polarity, it plays an important role in cell migration, cell polarity, cell shape, or both A and B. So it has a role in both cell polarity as well as cell migration is obviously a very default function of it. But cell polarity is also something that is coming about because of the microtubule polarity as well, right? Because it is maintaining the direction in which the centriole is present and the direction, or the centrosome is present and the direction in which it is getting extended. Then comes actin polymerization and branching. What does it require? So we saw the initiation and the polymerization that we quite in a lot of detail we looked at. So you saw that branching is taken care of by ARP23. And because I gave you some extra information as well, you saw that formin had a major role. It also had a major role, but here in particular, we see that ATP is said to be involved in actin polymerization as well. Then comes question 3, which is while lamins are being included, they are an example of which of the following: a type of microtubule, different structural organization of actin, an intermediate filament, or all of the above. They are obviously an example of intermediate filaments, and not actin or microtubules, of course. The lamins are, nuclear lamins in particular, are part of IFs or intermediate filaments. Then in question 4, we have that which of the following statements is not true? All the motor proteins have directionality. Microtubule plus end is pointed towards the plasma membrane. ARP23 nucleates new microtubule filaments, or microtubules are the central support in flagella. So we are being asked which of them are false. So motor proteins have directionality is true. Microtubule plus end points towards the plasma membrane is also true. The minus end is anchored in the centrosome. The plus end projects outwards. ARP23 nucleates new microtubule filaments is not true. It does nucleate microfilaments, but they are made up of actin and not microtubules or tubulin. And microtubules are the central support in flagella. Not necessarily, no. They are not the support molecules with respect to flagella. They are involved in the axonemal structure. Actually, that is true. They are forming the 9+2 structural subunit, which we see inside. Yeah, the question had asked for which of them were false, so that is not the correct answer. Sorry, I was a little bit disoriented. So option 3 is the correct answer for that question. Then we're being asked that which motor protein drives the bending of cilia and flagella. So we saw that dynein, like we saw that dynein present between the doublet, microtubule doublets is involved in the bending of cilia and flagella. So that is the correct option. The others are kinesins and myosins, which are obviously not correct. Then in question 6, we're being asked that which of the following statements about microfilaments is true. So microfilaments are the thickest class of cytoskeletal fibers. No. Microfilaments are made up of globular actin. Yes. They break under tension easily. No. And they are found only in cells that, or they're found in cells that lack microtubules. Also not true. So the only correct option here is option B, which is that they are made up of globular actin. So option B would be correct in this case. Then in question 7, we are asked that which of these cytoskeletal components lacks polarity, intermediate filaments, microtubules, actin filaments. So none of the above. We have discussed this very extensively, and you know the answer, the correct answer to this as well. So those would be intermediate filaments or IFs. Then the homologue of tubulin that is found in bacteria is MOR-B, CRE-S, FTSZ, or there are no tubulin homologues. Then, of course, the correct answer to that is FTSZ. And in question 9, we're asked that the role of motor proteins in cells is to activate cytoskeleton in the cell, move cargo vesicles in the cell, initiate microtubule and actin polymerization, or maintain the stability of microtubules. So motor proteins have the role of carrying cargo inside the cell, which is what they do using their tail domains as well. So both kinesins as well as kinase are involved in that job. Then in question 10, we're being asked that which of the following drugs inhibit microtubule polymerization. Nocodazole, vincristine. Nocodazole is not something I've mentioned in particular, but yeah, it's also one of the microtubule polymerization inhibiting drugs. Also vincristine and vinblastine are present. So all of the above would be the correct answer to that. So yeah, I think everything I had to cover for this week is done. If anybody has any doubts, please go ahead. Otherwise, I'll just go through the slides once for reference, and from there you can see if there is anything that needs to be brought up. Then we can go through that, and then we can end the class of today. Please interrupt me now if there is any doubt. Okay, if not, then I'll come back to these slides. Okay, yeah. Here you can see this is a inflorescence image. It is a falsely colored image. Most possibly, I think these are in mouse fibroblasts, and these are very beautifully imaged microtubules within the cell, which you can see, and actin filaments are propped towards the surface. And just a representative image of how intricate the cellular architecture is from within. Then this was the analogy that was given to Lego blocks, that you have individual sort of Legos or building blocks that are all together making up the cytoskeleton as we know of it today. Then there was a there was a video on actin filament assembly. We have looked at it also in the slides that we saw there about the proteins that were involved, how formin, ARP23, along with branching and other proteins which are involved in polymerization, in support, in cross-linking, etc. All of the different proteins that are involved, we looked at them. Then how the dynamic nature of cytoskeleton can be studied, how lamellipodi are involved in it. And in fact, there was a video here as well where you could see. Yeah, so there was a video there as well about how the movement of actin can occur inside the cells as well. And that was studied in detail. Then how I think there was a microscopic image of how microtubules and actins are moving with respect to each other. So since these are all video based, I'm not really able to show that in slides. But yeah, this was a sort of cross-section image of how actin filaments are arranged with respect to each other. If you remember, I had talked about how they can be arranged in the form of sheets or in the form of bundles. And here you can see that they have these sort of extensively branched networks. And there are proteins that help in the branching process as well. And that forms these very extensively, very intricately branched filamentous mesh sort of a structure that helps in projections, or that helps in giving it that structural support in projecting outside of the cell and in movement at large. And you can see the dimensions of it, that this is just half a micrometer, and within that you are seeing such huge number of actin filaments which are present, which gives you an idea about how small it actually is. And of course, you've read about the dynamic structure of it as well, that it has around 7 nanometers in diameter. So here you see the model for protrusion of the actin meshwork. You have ARP23, which is nucleating these filaments at 70-degree angles. The filaments are elongating, they're pushing the plasma membrane as a consequence forward. And when at a steady state, when the polymerized actin versus the freely available or the actin monomer pool, when they have somewhat become equal, no new subunits are added, and actin would end up in the ADP bound form. And this is susceptible to depolymerization by proteins such as ADF cofilin, which was also there in the, if you remember, in the slide that I had shown separately. So this is the disassembly which is occurring at behind the leading edge, and there is assembly which is occurring at the leading edge. So that's what we see there. Then come actin dynamics in philopodia. In the nascent philopodium, you are seeing there is, it looks like these are electron microscopic images. You can see how huge and how extensive the branching there is as well. So these are the actin assemblies or actin organizations that you can see inside the cell, and how the assembly is eventually resulting in a structure which is transient in nature. It's not a permanent complex, but there's a nascent philopodia which is being formed because of these particular proteins, and by the actin sort of reconstruction, which you can see there. Here you see the comet tail sort of structure, which is observed for Listeria monocytogenes. So you have actin, which is forming sort of a tail-like structure, comet tail-like structure, which are visible because of the arrangement or the assembly of actin at that particular place. Then come, of course, the cilia and flagella, which we also talked about, that microtubules become the central structural supports in their cases as well. So they are both able to move unicellular and multicellular, small multicellular organisms by propelling water using these structures. And if they are part of a larger structure, they can move fluid over the surface. Example being cilia, which is present along our respiratory tract, particularly within the lungs. The cilia, they help in movement of the trapped debris from the lung. So in fact, that is one of the defense mechanisms, or one of the innate immune defense mechanisms within our body as well, which protects us from a large number of pathogens. So that is another extended application that you can see in this case. And here you see how the doublet, I've already shown it to you, the 9+2 arrangement. Extensive details have been seen, but yeah, for now, here what you can see is that it is driven by dyneins, which are the major motor proteins working here. When phosphate group is added to from the ATP, when a phosphate group is added to dynein, it causes some conformational changes. They move the outer microtubules and the protein crosslinks. They remit the sliding, and instead of moving completely past each other, they are brought back to place, and there is a bending and then effective beating sort of movement which is executed. This is complex in just to just express it in words. The videos are much better examples of it. You can go on YouTube and you can exploit it as much as you want for your understanding of these biological phenomena, because you'll find a lot of wonderful animations which will help you understand. If you're somebody who studies better over text, of course, you can go to the reference books, but there is only so much you can visualize without actually getting into details about it. So these are the comparison of the three types of filaments, and you can see that it is actin is also extensively, or particularly just saying cytoskeleton, it is extensively in close contact with the plasma membrane and with the ECM as well. Then we talked about microfilaments, which are made up of globular actin, which has a twisted double chain of the actin subunits. They resist tension and they form a sort of 3D network just inside the plasma membrane. And coming back to the intermediate filaments, we talked about the fact that they are more structurally involved, or involved in more mechanical support. So they form structures like hair, nail, feathers, horns, etc. And intermediate filament genes are majorly conserved in metazoans, but fungi as well as plants, they lack these filaments. In fact, if you most Under normal conditions, cell culture that is very commonly done in most labs, they do not synthesize these proteins. They do not need to. So that is something that you can also look up if it is interesting to you. And intermediate filaments are believed that they have some sort of a metazoan linkage to them, and intermediate filaments got modified and were adapted for tissue as well as cell-specific purposes. They lack polarity. They do not bind nucleotides. They're oriented in opposite directions during that assembly process, and this leads to a similar structural appearance at both the filaments ends. And this was studied using FRAP, or fluorescence recovery after photo bleaching. And they have high tensile strength. They are resistant to compression, twisting, as well as bending forces. And we saw how the assembly occurs. They have much slower filament dynamics. They lack any sort of motor proteins. They do not, no such motor proteins till now have been identified to move along intermediate filaments, because they lack that polarity, and that assembly we have seen via a similar sort of structure. And you're seeing how these 8 tetramers are assembling in the rope-like filament that you finally see in the form of the 10 nanometer thick filament. 10 nanometer thick intermediate filament. And this, I think, is just the assembly which is being shown. Then came the idea of evolution of the cytoskeletal proteins. It was a feature that was unique to eukaryotes, and it was a revelation when it was first understood that there might be some related proteins within bacterial cells as well. They have homologs of both tubulin as well as actin in the form of FTSZ and MREP and protofilament axis, etc. You can see a comparison between the tubulin and the FTSZ as well as the actin and MREP. So there is some sort of derivative linkage which could be seen from the prokaryotes to the eukaryotes in this case. This is also a representation of how these individual structure might have been present in like the tubulin-like, actin-like, or intermediate filament-like structures were present across eubacteria and archaea and different subspecies along these classes as well. I'm not getting into details of it also. These will just basically recap the lecture otherwise. This you can see how these microfilaments are, microtubules are effectively involved in the cell structure. So you have the FTSZ, which is present during the time where there is an onset of cell division within S. aureus. How is the FTSZ arranged in the form of sort of ring-like structure, which has been probably visualized here using GFP, versus during elongation, or when there is in the advanced stages of cell division, you can see that it is leading to a sort of separation which would occur finally to give rise to two daughter cells. So these are just citations that are showing you that there would be some sort of an analogous structure present inside inside those cells as well. So yeah, I think these would be similar topics, the vesicular trafficking, which is being taken up by kinesins and dyneins. The mnemonic sort of which was given was for RAMP, which is retrograde, anterograde, minus, and plus. The plus and minus movement of the molecular motors themselves and how their directionality is looked at, what are the different proteins that are associated with them. So just if you look at this representative image for recapitulation, if we look at kinesins, they are moving towards the plus end. They are carrying the anterograde cargo. Anything that is moving towards think of it as something which is going in the straight direction, which is to move forward. You can attach a philosophical angle if you want to it, that you always should move forward in life. So anything that is moving forward is anterograde, versus something which is moving in a negative direction is retrograde. So that is in the opposite direction, taken care of by dyneins, cytoplasmic dyneins, and in assistance in some cases with dynactin, like I had also mentioned. So that would be a relatively easier way to remember the same as well. So this was the video that I was talking about. And here was a lot of extensive detail about the myosin superfamily for actin-based motor proteins. I don't think I went into that, so we can go over it here. So the First motor protein that was observed was the skeletal muscle myosin. Myosin 2 particularly was seen, which had two heavy chains and two light chains. It had that globular head domain that had the force generating machinery and very long amino acid sequences in that extended coil format, which was mediating heavy chain dimerization. So myosin head, it binds, releases or hydrolyzes ATP and uses this energy that has been released to move towards the plus end of the actin filament. It is an actin associated motor protein. And you can see that this is the myosin superfamily, which has a large number of different variants. And among these variants, you will see that the overall structure, if you just look at it briefly, the overall structure has a lot of variability, but it conserves certain domains. There is a largely conserved motor domain, and it has some sort of C-terminal region and N-terminal extensions, which can vary, but effectively end up performing the same functions. So many myosins can form dimers with two motor proteins per molecule, but some can function as monomers as well, depends upon the job that they are responsible for carrying out. Then you have myosin 5, myosin 2. Myosin 5 usually works as a dimer that processively transfers intracellular cargo along the actin filaments. And this is the non-processive dimeric myosin 2, which has similar functional domains, but it has a different form of assembly. Several proteins, they associate to the tail domains into filaments that can exert tension between the actin filaments. So, like I said, along the myosin family, different myosins will have different jobs. If you want, you can also go over and look at the different types of intermediate filaments, their different charges or their nature, and how that difference in nature is leading to the difference in their functional roles as well. Just give us an issue. Lead to less disturbance. Yeah. So that was myosin 5, then came myosin 6. It has overall structural similarity, but it moves towards the minus end instead of the plus end of the actin filament. And this is responsible possibly because of some sort of an insertion. When I say insertion, there is an inserted nucleotide most probably, or in other words, there's a mutation in its head domain, which is probably responsible for the change in its directionality. Then came the kinesins, which move around along microtubules, similar to myosin 2, structurally speaking. And we saw how kinesins can also move differently along microtubules as well. It is a plus end motor protein. So Saccharomyces cerevisiae has 6 distinct kinesins. Caenorhabditis elegans has 16. Humans have around 40. So what they do is that they move towards the plus end of the microtubule, and they carry a binding site at their tail for some sort of cargo, which can either be a membrane enclosed organelle or some sort of, or maybe another microtubule in general. Then there was the cargo docking, which was explained here. Not going into detail about it, slightly advanced concept, but if you want, you can actually refer to the papers as well. You can just go through Nature Reviews paper for how kinesin moves it and get into details about that. Here, I think these topics were already looked at, the myosin 4-5. Then came dyneins. These are minus end directed microtubule motors. They are unrelated to the kinesin superfamily. Their movement is in a completely opposite direction. Their structure is quite different, as you can obviously see here. Dynein has a much bigger head domain, which allows for much larger steps. When we say steps, we look at it as if a person is walking along the microtubule track. So therefore, the span that is covered by dyneins while they are walking is much larger. These are composed, this is the structure. As you can see, they are composed of 3 heavy chains, large number of light chains that are associated with it. And dynein majorly has 2 branches. Then you can see the 9+2 arrangement of the axoneme, the movement of these individual filamentous structures, comparison between kinesin, myosin, and dynein as well. And the debate upon unidirectionality and multi or bidirectionality, that was also something that was extensively covered in the lecture there. And I think, yeah, that kind of stops the presentation there. Yeah. So, okay. So I think I have covered everything I had to for now. Basically, everything that we looked at today, I went by a little fast. So if there are any doubts, please do let me know. But basically, all that we looked at was majorly focused around the cytoskeleton. We looked at each of the different cytoskeletal elements once again. We looked at the subtle differences between them, particularly with respect to how they are assembled, which is one of the major differences. The reason why there's a difference in polarity as well, how the nomenclature varies for actin, how it is a barbed and pointed end, how there's a difference in the assembly where it is ab initio compared to microtubules, which have centrosomes, in particular the gamma tubulin, in place for initiating that assembly, and how there are different types of proteins that are associating with these cytoskeletal elements in order to form the assembled 3D structure. We looked at ARP2, 3 cofilins, profilins, formins, and so many other proteins, tropomyosin, tropomodulin, some branching proteins, etc. So that gives you an idea about how large and how extensive this entire structure is and how we know so little about it till date. Then we moved on to intermediate filaments, the examples of those, and then we eventually moved towards microtubules, which while we have already discussed, we went into a little more detail about it this week. So how the microtubule structure is made up of, how it is made and how the arrangement of the microtubules in a different manner can lead to an entirely different structure. I mean this with respect to the axoneme, which is the basic structure that you would find inside cilia and flagella as well. So in that case, you are seeing that there is an incomplete sort of doublet, which is also getting formed, that there is one filament which is complete with 13 protofilaments, and there are other half sort of filaments or incomplete sort of filaments, which are made up of 10 or 11 microtubules, which are assembling one around each other. So that gives you the assembly of the 9+2 arrangement, and how it is linked to a molecular motor, such as dynein, is executing the movement that we talk about. If you want, you can go into details about the sliding filament theory as well. I'm not sure how relevant it is at the moment, but for your own understanding, you can. I did not include it in the slides because that is itself quite a big topic. But if it is something you want, I could compile a slide for it as well. You can mention that. So yeah, those were the major topics. We look at the different types of molecular motors and why they are so relevant is something you'll probably get to know more about if you look into vesicular trafficking and the way the transport plays such a huge role in this trafficking process. So till now, what you have as are the tracks on which the trafficking can be carried out. That trafficking has to be carried out by certain proteins, some of which are, of course, these kinesins, dyneins, and there are other proteins as well. But the regulation of that trafficking is also so intensive and it has to occur correctly. Otherwise, it can lead to n number of diseases. Like if a particular protein that is supposed to be present in the lysosome is going somewhere else, but because of which the lysosome is no longer storing that protein, that leads to its disease, a very, very serious disease. Like there are lysosomal storage disorders. So all of these processes, I have said this multiple times, but all of these processes that occur in extensive coordination with each other, and they execute the function of a living cell so seamlessly that we don't even seem to realize how it is occurring. But yeah, we are getting some insights into it in these topics for cell biology. So I think that would cover everything I had to say for today. We will be looking at the topics for next week in the next session. For now, if anybody has any doubts, please go ahead and mention them. We'll try to clarify those. So please unmute yourself and go ahead. Okay, if not, then you can, I guess we can wrap up the session for today. And if anybody has any doubts, you can bring them up in the next class, or you can mention your doubts somewhere on the discussion forum as well. And I hope today's session was helpful for you, and it would help you solve whatever questions are coming up and in the exam in the long run as well. So thank you so much for joining today. Have a very nice evening and all the best. Thank you.